All right, welcome to Group 1's final presentation for our lab, for BioLab 2. This is the variations in population with wood lice experiment. My name is Megan Reynolds. I am presenting the intro slide as well as the introduction. Kayley Ross will be discussing the methods. Samantha Beamer is discussing the results, and Tyler Bracewell will discuss the discussion. I also apologize if you hear any whining in the background. That is my puppy. So let's just get started then. So for the introduction, I'm going to start by discussing the environment, some selectivity, as well as our purpose. So I'm going to start with some general information. So the wood lice are typically found in hot, humid environments under rotting logs or rocks. And the sprint speed is highly selective in both survival and reproduction. And our purpose for this experiment was to test the sprint speed of wood lice in varying conditions. Our group decided to test if having food present at the end of the track affects the sprint speed of the wood lice. So I'm also going to discuss the stress study, goal, hypothesis, and citation. I'm going to really quick make a note here on the citation. We found a similar study known as rotational stress, and it was written by someone named Anselme from 2018. And I'm going to just go a little bit into the similar study here. So in this study, the rotational stress it was quoted, the rotational stress influenced sensitized but non-habitualized exploratory behavior in wood lice. And the results showed that, in quotes, wood lice were more and more inclined to escape from a hostile environment where no food was available. So based on this information, our group wondered how would food affect the sprint speed. So our goal of this presentation, of our experiment, was to find an answer to our question, which was, will the presence of food affect the sprint speed of wood lice? So after using the similar studies and knowing the general info of the wood lice, we formulated a hypothesis, which was that the presence of food will increase the sprint speed of the wood lice.
The methods section of our experiment. For our experiment, we studied wood lice. We split our wood lice into two groups, a treatment group and a control group. The treatment group was wood lice in the presence of food, while the control group was wood lice without food. The experiment was completed with a sample size of 20. This sample size was spread out over 4 different study sites. Each group member had their own study site at their own house where their portion of the experiment was completed. Each group member had 5 wood lice. An example of a study site is shown on the next slide. This was the study site used by Sam Beamer. The materials used included cardboard that was used to build a track that was 20 centimeters long and 5 centimeters wide, also as shown in the previous slide. Dried leaves were used as the food for the treatment group. The controlled variables included the food, the length of the track, and the track material. The experiment began by holding the wood lice for 2 days. This allowed us to make sure that they ate the dried leaves we were going to use for food. The food was then removed the night before the trials in order to make sure the wood lice were hungry. 20 trials were then completed. In the trials, 10 were performed with food at the end of the track, and 10 were performed without the food. The time it took each wood louse to run the 20-centimeter course was recorded along with the wood louse's body length. The process of running the course was repeated twice for each wood louse. The statistical test performed was a 2-sample t-test. It was used because it allows us to compare the means of 2 groups. In this case, the means of the control group and the treatment group were compared.
In order to compare the adjusted sprint speed of wood lice with food to wood lice without food, we decided to perform a 2-sample t-test. We chose a 2-sample t-test because a 2-sample t-test is used to measure the average of 2 different means, which is exactly what we wanted to do. We found the average sprint speed with food to be 0.186 and the average sprint speed without food to be 0.435. We then performed a 95% confidence interval and found that we are 95% certain that the mean sprint speed for wood lice with food is between 0.145 and 0.227. We also have found that we are 95% confident that the average sprint speed without food is between 0.187 and 0.683. This graph is just a graphical representation of the data shown in the previous slide, and as you can see for the control group, there is a lot more variation in the data than there is for the treatment shown, as shown by the error bars. The control group also has a higher average adjusted sprint speed than the treatment group, meaning there may be a significant difference. So we then decided to perform a 2-sample t-test, and these are the results. Variable 1 is the control group, and variable 2 is the treatment group. And the second from the bottom row shows that our p-value was found to be 0.05. And since it was equal to 0.05 and was not less than it, that means that there is not a significant difference in the sprint speed with food to the sprint speed without food.
So part of what inspired our interest in seeing if there would be an effect on woodlice sprint speed based on food was a study done by Ford and Shuttlesworth back in 1986. They did an experiment where they tested garter snake sprint speed based on how much these snakes had eaten before the test. This amount of food was based on the snake's body mass, and they found that a starved snake sprinted faster when chased. Our experiment involved using starved woodlice, whereas the experiment done with garter snakes was done using various states of satiety, so our test was using the food more as a lure. When analyzing our data, we obtained a p-value of 0.0516, which is greater than the accepted value of 0.05, so we have to reject our hypothesis that the presence of food at the end of the track would have increased the sprint speed of the woodlice. And this is further supported by our graph because of the overlapping error bars, which represent the 95% confidence interval. Doing some research after our experiment helped us to find another study done by Kayley and her colleagues. They looked into different factors that would affect woodlice sprint speed, like temperature, desiccation state, and body mass. The important takeaways from their experiment were that a higher temperature led to an increased sprint speed, while a lower body mass led to the opposite effect. This could be related to desiccation state as well as the amount of food the woodlice eat, which is the relation to our experiment. After reading about the experiment, it brought to some light some difficulties that were hard to control in our experiment. These variables being the temperature, since the 4 of us were in different locations and doing the experiment at different times of day. The population of woodlice was also out of our control because of COVID-19 and our inability to collect woodlice from the same source. This means that the different woodlice tested could have different reactions to food. Though we had these difficulties, the errors we had could potentially support the data from Kayley and her group. If we were to go forward with the data we currently have, then the population name would have to be adjusted and called a Northeast Ohio population to cover the different woodlice collected by the 4 of us. Some improvements for the experiment would be to use the same population of woodlice to prevent any differences from occurring based on that, like food preference and familiarity to each other. Also, deciding on a day and time for the experiment would help to minimize the effects of temperature and humidity, and from humidity, therefore, the desiccation of the woodlice. Also, deciding on the state of decay for the food would help to standardize the attraction to the food, to then consider it as a new variable in a later experiment. For other areas of future research, we could look into the wider range of foods that appeal to woodlice, as well as if there are any different preferences for how decayed the food is when presented to them. The amount of food presented at the end of the track could also be an influential factor to investigate, to see if the woodlice would respond to that, and if that would therefore increase their sprint speed. Also, investigating the hunger state of the woodlice based on how many hours or days they have gone without eating could affect their recorded sprint speed.